As Sean mentioned, I'm Ruth Rivkin. I'm an undergraduate at the University of Guelph. And today I'm going to be discussing the mechanisms which contribute to the persistence of polymorphisms in nature. I'd like to begin with a question. How are polymorphisms maintained? Polymorphisms occur when there are two or more phenotypes which exist for a single trait within a population. The most well-known polymorphisms have to do with color. A good example of this is on my right. And these guppies here, although they are the same species and even the same gender, there's a wide variation in color patterning, and this variation exists throughout nature. However, polymorphisms should be eliminated through directional selection, because one phenotype should do better than the others and should spread to fixation within the population. So, polymorphisms, we need another explanation, and that can be offered through frequency-dependent selection. Under frequency-dependent selection, the fitness of an individual depends not only on the phenotype of that individual, but also on the frequency of that phenotype within a population. There are several modes of frequency-dependent selection, and the one which best explains the maintenance of polymorphisms is negative frequency-dependent selection. Under negative frequency-dependent selection, an individual's fitness should decrease as the frequency of its phenotype relative to others increases within the population. So an individual will have greatest fitness when its phenotype is at low frequencies within the population. This is called having a rare fitness advantage. In our lab, the polymorphism which is of greatest interest to us is gynodioecious, a sexual polymorphism in which hermaphrodites and females co-occur. Gynodioecious is thought to be a transition state between pure hermaphroditism and dioecious, the co-occurrence of females and males. In gynodioecious populations, hermaphrodites produce both pollen and ovules and should be able to persist in the absence of females, who only produce ovules. In contrast, females require the presence of hermaphrodites because they provide the pollen for ovule fertilization. And this leads me to my second question. How are females able to persist in the face of this competitive disadvantage? That can be explained by negative frequency-dependent selection, because females should have a fitness advantage when they are rare in a population. Females should have this fitness advantage through variation in the availability of pollen. In gynodioecious populations, hermaphrodites are the sole producers of pollen, and as a result, the availability of pollen is entirely dependent upon the proportion of hermaphrodites within the population. When a plant does not receive sufficient pollen for ovule fertilization, its fitness is decreased because it cannot set as many fruits and seeds. When this happens, the plant is said to be pollen limited. In gynodioecious populations, as the frequency of females increases in the population, the availability of pollen should decrease, and female fitness should be reduced. However, although variation in pollen availability is frequently proposed to be the mechanism of negative frequency-dependent selection, it has not been properly experimentally tested. And that's what we decided to do. Based on this information, we had two questions. First, could negative frequency-dependent selection be contributing to the persistence of females in gynodioecious populations? And second, is differential pollen availability the mechanism of negative frequency-dependent selection? To answer these questions, we use Lobelia siphilitica. Lobelia is a gynodioecious wildflower which has impressive variation in population sex ratios. In fact, natural populations have been found to have anywhere between 0 and 90% female frequencies. Lobelia is a bumblebee-pollinated species, and although self-fertilization does occur, it is rare because the male and female phases of hermaphroditic flowers are temporally separated. Thank you. So we created 4 arrays containing 20 plants within each. To test for frequency-dependent selection, we varied the sex ratio of each array from 20, 40, 60 to 80% female. We also hand-pollinated half the females within each population to test for the influence of pollen limitation on frequency-dependent selection. The fitness of hand-pollinated females should not be correlated with population sex ratio because we manually applied pollen to the stigmas of each receptive flower on the selected plants. This means that the hand-pollinated females should not be pollen limited. We replicated each treatment 8 times within 4 blocks, and we assessed 2 fitness measures, the number of fruits set per plant and the number of seeds set per fruit. Before I take you through our results, I'd like to just go through the predictions which we set prior to starting the experiment. We predicted that the fitness of females would be frequency dependent, such that the fitness of females would decrease as female frequency increased in a population relative to hermaphrodites. We also predicted that if the availability of pollen influences frequency-dependent selection, then fitness will decline only in openly pollinated females. If this is the case, then we expect the fitness of open-pollinated females to be frequency-dependent and the fitness of hand-pollinated females to not be correlated with population sex ratio. We also expected that the fitness of hand-pollinated females would have higher fitness than openly pollinated females. We found that total seed production was indeed frequency dependent. And before I go through the rest of this, I'd like to take a moment to walk everyone through my graph because several features will occur again in future graphs. So the data presented here is raw data and it's not least square means. Each individual point represents the average across a single treatment group within a replicate. Total seed production per plant was determined to be the product of the number of fruits set per plant and the number of seeds set per fruit. So we found that as female frequency increased, total seed production decreased significantly. In fact, when females were rare in a population, they set 30% more seeds than when they are common. We also found that total seed production was varied between pollination types. In this figure, total seed production is on the y-axis and pollination type, hand versus open, is on the x-axis. We found that openly pollinated females set 54% fewer seeds than did hand-pollinated females. Despite this, we found that total seed production declined regardless of pollination type. And this figure shows the interaction between pollination type and array sex ratio. And total seed production is again on the y-axis, female frequency on the x-axis. I'd like to draw your attention to 2 things. First, there was no significant pollination by sex ratio interaction. And secondly, although we had predicted that the fitness of hand-pollinated females would not be correlated with population sex ratio, we found that total seed production declined as female frequency increased in both hand and open pollinated females. This was highly surprising because we found that the relationship between total seed production and female frequency was almost identical between pollination types. To recap, I'd like to bring you back to my question slide. So we did find evidence that fitness was frequency dependent, and this suggests that negative frequency dependent selection could be contributing to the persistence of females. However, we did not find evidence that differential pollen availability was the mechanism of negative frequency dependent selection. Instead, we found that flower production decreased as female frequency increased. In this figure, mean flower production is on the y-axis and female frequency on the x. We found that when females were rare in a population, they set 26% more flowers than when they are common. We also found that the availability of pollen did not influence flower production. This figure here shows the interaction between pollination type and sex ratio. And I'd like to again point out a couple things. First, there is no significant interaction between pollination type and sex ratio. And second, flower production decreased as female frequency increased in both hand and open pollinated females. This was highly surprising because although it is clear that females are upregulating their flower production when they are rare in a population, the trigger of that is clearly not the availability of pollen. We do have a few ideas as to what could be contributing to this upregulation. However, from this data set, there's no clear explanation. Thank you. What we do know is that flower production can regulate the fitness of a plant. This is because a plant can only produce as many fruits and seeds as it has flowers. So if a female is producing more flowers when she is rare in a population, she should also be setting more fruits and seeds and should have higher fitness than when she is common. So in summary, we found that variable flower production and not pollen availability was the could be a mechanism of negative frequency dependent selection. And flower production is a newly proposed mechanism, and although the underlying causes of it are not currently known, we do know that this new mechanism really expands the conditions under which negative frequency dependent selection can take place. This is because when a female should maintain her rare fitness advantage even when pollen is not limiting. Our results suggest that gynodioecious may be a more stable polymorphism than had previously been anticipated. And finally, I'd like to leave you with this last thought. Pollen limitation is assumed in the literature to be a key mechanism of negative frequency dependent selection. However, until our study, it had not been accurately tested. We found that pollen limitation was not a mechanism of negative frequency dependent selection, and this really highlights the importance of testing the mechanisms of selection which are proposed in the literature and not just assuming that these mechanisms are correct. I'd like to thank everyone who contributed to this project and gave advice on the writing and presentation of it, and of course the funding services and the audience for listening, and I welcome all questions.
Thank you, Ruth. We have 3 minutes for questions.
Okay, thank you very much.
What about the hermaphrodites? Because if they do not self-pollinate that much, they should also have a lower seed production when you have a lot of females in your crop.
Yeah, so the question was about what do the hermaphrodites do if they're not self-fertilizing, then they should also experience frequency-dependent selection. So I actually have a few bonus slides showing the hermaphrodite data. I'm going to go to fruit production. And this graph is a little bit confusing, I apologize, but the triangles are female and the circles are hermaphrodite. And open circles mean open pollinated females, and this is a little backwards, open pollinated triangles mean hand pollinated females. Anyways, so what we found was that hermaphrodites showed no sign of selection. They were pretty much consistent right across the board. There was a slight increase in fruit production as female frequency increased, so as hermaphrodites became rare. However, this difference was not significant, and we also see this in the hermaphrodites for flower production. Oh, sorry. So again, this line is almost straight, and there's no significant difference. In the back?
So I think it's a little far-fetched to say that this doesn't happen in nature. It's a highly contrived experiment. I think we need to be a little bit cautious about overstating that. That said, should try to think about the reason for why you got this result. And I agree, the flower production data is totally unexpected that females' flower production went down. So my question is, were you careful to control the number of families that were represented in the different arrays? Because by decreasing or increasing females or hermaphrodites, you could be changing the number of families, and you could be changing the number of sires, and maybe it's related somehow to that.
So we did control for family members within the hermaphrodite populations to try and avoid inbreeding depression. So we had no more than 2 family members within each population, and in the low frequencies for the hermaphrodites, we had only 1 family member within each.
And when you did your hand pollination, how many different families did you use?
We had donor hermaphrodites and used a variety. It was always mixed pollen. Yep. In the back.
I was curious about whether you saw any differences in floribor attraction to the cross given the different frequencies of hermaphrodites versus females.
So
things that eat flowers.
in pollinator attraction?
No, floribor, so things that eat flowers or pollen.
Okay, we tried to avoid the big ones like groundhogs and deer. We had them fenced off. We didn't look at herbivores, so the insects.
Yeah.
But just from personal observation, there didn't seem to be too much floral damage. But again, we didn't really look at that.
One more quick question.
What are the natural habitats of this plant? And I know this experiment was done in sort of open environment, sort of controlled as well. So the number of bees versus that can be the natural versus the artificial, the made environment can be different. So did you quantify that?
So the natural habitat of Lobelia is sort of grows in ditches and wet areas, and we did water. I was out there every day watering to control for that. The bees, we were out in a center that did have a few beehives, or that did have an abundance of bees, so I don't think they were pollinator limited in any kind of way, although again, we didn't really quantify that.
All right, thanks again, Ruth. Great.